Hello everyone, welcome to the 26th edition of our INFOGEST Viva Webinar. My name is Nikki, and I would like to introduce my colleagues Sarah Perez Villa and Shannon Guala, who will conduct the webinar with me. This event is organized by a team of postdocs from Teagasc Food Research Centre in Ireland. The webinar series is designed to showcase the latest findings in digestion, featuring presentations from recent PhD graduates, postdoctoral researchers, and early career scientists. The presentation in a pre-recorded video will be followed by a question and answer session with the speaker. So I invite you to use the Q&A button at the bottom of your screen. Our speaker today is Nicholas Horlacher, with his project about post-electric field pasteurization, followed by probiotic fermentation, alters the in vitro protein digestibility of plant-based dairy alternatives. Nicholas is a recent PhD graduate in food science from the University of Otago, where he worked under the supervision of Professor Indrawati Oey in collaboration with the Riddet Institute. His research explored the potential benefits of pulse-electric field technology combined with lactic acid fermentation as a novel processing strategy in dairy alternative applications, focusing on protein-rich plant-based milk and yogurt. Now, let's hear from Nicholas.
Good afternoon, and thank you for joining this INFOGEST webinar. My name is Nicholas Horlacher, and I'm a recent PhD graduate from the University of Otago. And I'm quite happy today to share part of my PhD project that explored the effects of pulse-electric field processing on the nutritional and functional properties of plant-based dairy alternatives. And so this project was funded by the Riddet Institute here in New Zealand with Indrawati Oey as my primary supervisor and in collaboration with Plant and Food Research. And so I'll start with a brief introduction. So pulse-electric field, or PEF in short, is a new technology that has been explored since around the 90s as a potential replacement for conventional thermal methods in various areas of the food production chain. And so as a key benefit, it's electricity-based and so does not rely on the use of fossil fuels, and also shows a lower energy consumption, which makes it quite an interesting environmentally friendly option. And in liquid food products, PEF is mainly used for the purpose of pasteurization, where it can achieve the required microbial inactivation at lower temperatures than thermal processes. And so it's often used as a mild pasteurization to preserve thermally sensitive nutrients. And PEF operates by applying several high voltage pulses in the microsecond range, which can cause the formation of pores in bacterial cell membranes, resulting in their bacterial death. A process that's also often referred to as electroporation. And in the application in dairy alternatives, the use of PEF is still quite new, but it's clear that other food constituents are also affected at the same time. And so this includes the product's protein contents, which can undergo a partial unfolding and reaggregation process based on applied treatment intensities. And so previous work actually showed here that this can be used to achieve a positive impact on important protein functionalities such as solubility, emulsification, foaming, which dictates the quality of plant milk and yogurt products. And so that brings quite an interesting added benefit to the use of PEF. And so digestive properties are, however, known to also be affected by microstructural changes of protein. And so there's a remaining question that hasn't been answered to date on how PEF can actually impact the protein digestibility and by extension the nutritional value of plant milk and yogurt products. And based on the essential role that high quality dietary protein play in supporting human health, this is quite a significant issue to examine when considering the use of PEF in food processing. And so the overall goal of the research was to evaluate the impacts of PEF on plant protein when applied alone on a plant milk type product, and then also when applied in sequence with lactic acid fermentation on a yogurt type product. And to achieve this, I actually started by developing a model food matrix where OAT and P were selected as the main base materials. Mainly that's because of their complementary amino acid profile, which helps to improve the protein quality of the milk, whereas pea protein also possesses significant gelation properties that can be used to create textures that resemble regular yogurt, And so it's quite a suitable formulation for this product type overall. And as a first step, microbial inactivations during PEF were studied to identify process parameters that qualify as a pasteurization by permitting sufficient inactivations of microbial cells. And once this was complete, I applied a lactic acid fermentation step in sequence with PEF to explore the effects on protein digestive properties of the two product types. And so this is the part that I'll mainly focus on today. And so this shows the overall study design that was used. And so I go through this step by step, starting with the sample preparation. And so to prepare the model food, firstly, oat flakes were hydrolyzed, filtered, and then mixed with a dissolved pea protein at a ratio of 1:1, which resulted in a product that contained protein at about 5%. And the plant milk was then pumped continuously through the process assembly at a constant flow rate of 14 liters per hour. And so firstly, it went through a preheated coil that was submerged in a heated water bath. And so this step aimed to precondition the product to temperatures between 30 to 45 degrees, which helps quite a lot to improve the effectiveness of PEF to achieve microbial inactivation. And so then the product was passed through a single collinear type PEF chamber where it was exposed to bipolar square wave pulses and an electrical field strength of 10 kilovolt per centimeter. And based on the applied pulse frequency, the specific energy input can also be calculated, which was used to define the intensity of the treatment. And so I'll refer to this throughout the talk. And then following rapid cooling after the treatment, plant milk samples were collected, while the remaining product was also subjected to fermentation treatment to also prepare the yogurt alternative. And then to assess the protein digestibility of the two food types, the INFOGEST static in vitro digestion protocol was used. And so that involved a brief oral phase at pH 7, followed by a 2-hour gastric phase and a 4-hour intestinal phase, where sampling was done at multiple time points, so a total of 13 time points throughout the digestion. And then for analysis, the digested protein material was extracted from each digester using TCA precipitation and then quantified by the OPA method based on available alpha amino group contents, with lysine was used as a standard here. And then alongside acid hydrolysis was used to determine the total amount of alpha amino groups in each sample, which that actually allows to quantify the degree of protein hydrolysis as a function of time. And so that was also used for kinetic modeling to determine the kinetic parameter of the digestion. And as a separate analysis, amino acid profiles were also determined with the goal to identify the presence of limiting amino acid contents by comparing contents to a reference pattern for children and juveniles. And so combining those two results for protein digestibility and the limiting amino acid score could then actually be used as an in vitro approach to estimate the overall protein quality of the product. And lastly, as contributing factor to antinutrients, namely phytic acid and trypsin inhibitor, were quantified, since they also have a big impact on the digestibility of most plant-based foods made from materials such as oats and pea. And so then I'll start with the results and discussion. So there's actually two separate studies that I can share today. So the first one, we used a single PEF process, so a single set of process parameter were applied to pasteurize the plant milk, which was then followed by fermentation with a range of selected organisms. And so these, the top two are quite established probiotics, which bring interesting additional benefits when used as a starter culture in plant yogurts, whereas the bottom two are two conventional yogurt cultures that are also used commercially at the moment. And so this graph now shows the results for the gastric digestion phase. And so a zero-order model was applied here, which showed quite a suitable fit, which firstly shows that the digestion overall was limited, reaching only about 10 to 12% after 2 hours. And also there was a limited variation between control and PEF, and also fermented samples followed quite a similar trend. And so that shows the overall impact of processing on the digestion rates by pepsin were quite limited here. And so going to the intestinal digestion in contrast, we can see that firstly, most of the amino acid material was released actually during this phase. And so here we used a fractional conversion model to describe the kinetic increase over the 4-hour digestion time. And this actually shows that there was a more tangible impact by the applied PEF treatment. So especially after about 1 hour in the later phases of digestion, we can see a separation between the two. And when we apply PEF and fermentation together for the preparation of yogurt alternatives, we can see that there was also further increase in digestibility for all 4 of the selected cultures. So that was quite an interesting finding to start. So the next step was then actually to take a look at changes in amino acid contents. And so we can look at the first column for the control, where we can see that all of the amino acid scores were actually above 100, which shows that combining of oat and pea ingredients in the study was actually quite successful at avoiding the presence of a limiting amino acid. And applying PEF also showed no significant influence here, so as expected, degradative effects that can take place during thermal pasteurization treatments were limited during PEF. And when applying fermentation as a next step, we could, however, see several varying effects. So few amino acids, such as those examples here, were also not affected, as we can see. So there was slight variations, but not significant statistically. And so when it comes to other amino acids, such as threonine, we could, however, see a significant increase, whereas others also decreased significantly. And so that's not that unexpected. Related to the metabolic abilities of the selected cultures, we could expect that some amino acids are consumed to support the growth of the organism, while others are also produced and as such accumulate in the milk. And so in the case of the sulfur amino acids, which include cysteine and methionine, this, however, could result in scores below 100, which indicates that the nutritional quality of the yogurt could actually be impacted by the fermentation. And so combining these results, so the lowest score from the amino acids, together with the digestibility endpoint, also gives us then an indication whether or not the overall nutritional protein quality was affected. And so that's depicted in the figure here, where we can see that there's actually a general increasing trend. So we could consider the use of PEF and fermentation as beneficial for the nutritional benefits of the yogurt alternative, which showed also a dependency on the applied starter culture. And so that's quite good so far, but it's actually still unclear whether this effect can be attributed more to the effects of PEF or fermentation, and so which treatment was actually the bigger driving factor for the results of the combined processing was a big question we had at the time. And then in addition, the question that's also important to address is how any temperature changes during the PEF process might have impacted and played a role in the results. And so to probe this further, we actually used a second study. And so here we assessed in vitro digestion as a function of PEF process parameter, namely the preheating temperature and specific energy input. And a second control was also included where we fermented the plant milk without applying PEF. And so to start, I'll share the intestinal digestion results of just a single PEF treatment. So firstly, when applying PEF on the unfermented control, we can see an increase in digestibility, which further increased. Further increased in PEF. And then also PEF and fermentation showed a further increase quite in a similar way to the previous trial. And so that actually also shows the reproducibility of the findings, which is quite good. And so looking at the kinetic parameter on the right, we can also quantify this increase, which shows that mainly the digestion endpoint was affected, where you can see there's a non-overlapping confidence interval between the control and both of the PEF treatment samples, which also indicates that there was, in fact, a significant effect. And so, but when we include the yogurt that was prepared without PEF, so the second control, we see that there's actually quite a limited impact here. So that's the dashed line that almost overlaps towards the end with the control. And so that shows that fermentation alone actually didn't seem to affect digestibility very much. And so this figure also shows just focusing on the digestion endpoint of all of the samples as the next step. So mainly here we can see that the effects of PEF seem to increase as a function of the applied process parameter. So going from the left to the right, there you can see an increase. And looking at the lowest treatment intensity, both fermented and unfermented samples had very similar results. But by increasing either of the two parameters, so the preheating temperature or also specific energy input, the digestibility increased at the same time. And so overall, this suggests that there was actually a synergistic influence by PEF that enhanced the digestibility of the plant milk, but also influenced the effect of fermentation on the plant yogurt. And so this table now shows the product temperatures that were also measured during PEF. And so we can see, I indicated with green here, several treatments that could be considered in the milder region, where temperature increases due to Joule heating that take place during the treatment were limited. And so those also showed an impact on plant yogurt digestibility, which is interesting. And considering that the thermal denaturation point of the plant milk protein, which was mainly legumin and viscellin coming from the pea isolate, is around 70 to 80 degrees, we could conclude that the influence of PEF was likely attributed to a direct effect of the applied electric fields, whereas at the higher intensities, the heating effects during the treatment should be expected to contribute to the results alongside electric field effects. So that would be a combined impact of the two. And so I'll finish by talking about the anti-nutritional contents. So starting with trypsin inhibitor, that could also be found in the plant milk and likely influence the protein digestibility. And so we can see that there was a partial degradation based on the treatment intensity that also correlated quite well with the digestion endpoint. And so that suggests that PEF achieved a structural change in an anti-nutritional protein, which inhibits the binding with trypsin and as such enhances the digestibility. And the second antinutrient that I looked at was phytic acid, which can also impact protein digestibility by forming ternary protein complexes that show a reduced susceptibility to trypsin digestion. And so in this case, we can see that fermentation had a significant influence, but combined treatments were also more effective, which further displays the synergy between the two treatments. And so overall, this shows that PEF and fermentation likely influence protein digestibility by partial degradation of antinutrients, where PEF also played quite an important role in the overall effects on the nutritional quality of the product. And so finally, the blended oat and pea type formulation was overall quite suitable to prepare a protein-rich dairy alternative, while applying PEF and fermentation together can be considered beneficial to at least partially alleviate the low digestibility of the resulting product. And in addition, the findings here today show that there was also potential synergy between PEF and fermentation that contributed to the observed results, such as through a partial degradation of anti-nutritional contents. And so that concludes my talk for today. So thank you for your attention. And if you're interested in PEF or other processing technologies as well, there's actually quite a good YouTube channel that I can refer you to that was started by my primary supervisor. And there's also a few publications that come out of this where you can read a little bit more about the research. So thank you.
Thank you so much, Nicholas, for the really nice presentation. It was really interesting, and now we will move to the questions and answer session. Um, well, I will start with one of the questions I had. And did you observe any additional impact of the pulse electric field on other qualities apart from the digestibility of the samples, like other aspects like the sensorial properties or the functional properties of the proteins like solubility?
Yeah, so there definitely was quite a lot of changes. So in the talk, I'm mainly focused on digestibility now, but as part of my PhD, a big focus was actually on the functionality of the protein as well. So I looked at the protein solubility, for example, which we found it tended to increase, which is quite interesting because it affects, for example, the sedimentation of plant milks, which is a common
Mm,
problem. So
yeah.
it reduces the amount of material that settles to the bottom and sort of forms a sludge, which can be a quality defect. But it could also relate to the digestibility because more soluble protein should be more accessible to enzymatic digestion. So the two may be very well related as well.
Mm. And like you were studying in the blends of the oats and pea protein, but do you think that will be an effect on the proteins separately or more effect in one of them than in the other?
Yeah, that's an interesting question, actually. So it's quite difficult to determine because I only looked at the mixture of the two. We did use FDAR, which showed that the pea protein were mainly the dominant part, so there was a lot more pea protein than oat protein present as well.
Okay.
And also pea protein tend to be more susceptible to changes, so they have a much lower denaturation temperature compared to oat protein, for example. So yeah, based on my opinion, I think that should be mainly based on the changes in pea protein, and oat protein might be less affected, but it's difficult to say that
Okay.
definitively. But yeah.
Yeah. Well, there's a question from the audience. They
Mhm.
ask, did you analyze inhibition in every phase of your digestion, and what's the impact of other inhibitors on protein digestibility?
Sorry, could you repeat that
Sorry,
question? Mhm.
did you add enzymes inhibitors in every phase of your digestion, and what is the impact of other inhibitors on the protein digestibility?
Yeah, so because we only looked at the protein digestibility, I mainly focused on the trypsin inhibitor.
Okay.
I think chymotrypsin inhibitor often they're looked at together, so I would expect that there would be an impact also on chymotrypsin inhibitors,
Yeah.
which would also affect, so likely that there were a similar impact on the two. But yeah, I only looked at the trypsin inhibitor in this.
Um, which method did you use for in vitro protein digestion calculation? Is a question from Anosri.
Yeah.
Um,
So what we did, I used a static digestion method and collected time points over time. And we stopped the digestion with TCA. So TCA to denature the enzymes and precipitate protein. And as a reference, we also did a acid digestion where, yeah, so acid hydrolysis. So I basically compared the digester divided by the reference, the results for the reference sample by acid digestion
mhm.
to get the
So
percentage.
super. Thanks, Nicola. Well, we have a question from Hiba. She's asking about the sensory evaluation of the products. In case that you could tell us a bit more about that part of your work.
Yeah, so I tried it myself, which doesn't really count as a sensory trial, but based on the scope of the project, we didn't include any sensory evaluation. So I mainly focused on the main focus of the whole program was on determining the impacts on the protein contents as well. So I think it would be quite interesting to go a step further and actually look at the overall impact on its sensory properties.
Okay.
But
And
yeah, didn't
sorry.
include that in there. Yeah.
From Verena, we have a question that she's asking if did you look into the effect of the pulse electric field treatment on the micronutrients, like for instance vitamins? I don't know if you look into that or
No, so
no.
unfortunately,
Okay.
also that's a very big focus. Other papers that looked at PEF treatments on fruit juices and things like that as well. So we mainly focused on the parts that no one has done so far.
Mhm. Okay. And did you know how the formulation, like the oat protein and pea protein ratio, the solids content of the formulas and all those affect the effectivity of the pulse electric field?
Yeah, that's an interesting question actually. So I would expect that if the oat protein content is higher, then the impact might be reduced, just based on how more resistant oat protein are to processing in general. So the more pure pea protein milk, for example, might have even a bigger impact from the processing. But
And we
yeah,
have
so
a last question from Hiva, and she would like to know your opinion. So this is an open question. Which is your opinion on the pulse electric field and fermentation that could have in other legumes, like she's asking specifically about lupin. So I don't know if you had an experience about that before.
or like a more general
Yeah,
opinion on the processing.
I think so.
Yeah, so to me, I think it's quite that have in general the main interest is the sustainability aspect, because it's often looked at as a replacement to thermal processing, which is interesting because they can use directly renewable energy as opposed to fossil fuels, which are still very common in use in thermal processing. So gas and coal is mostly applied, which PEF can basically give away to avoid those.
Super. Thanks a million, Nicholas. It was
Mhm.
it was great to have you presenting your work on digestibility, and really interesting topic, really interesting presentation. And thank you so much for answering all the questions we had from the audience. So, well, now I would like to introduce that we will have next speakers for the following months. And it will be Marina from the Technical University of Berlin and Francesca from Teagasc Park. We are open to receive new expressions of interest if any of would like to present their work on digestion. They are welcome to the email of the INFOGEST Viva. And well, thank you so much, Nicholas, once again for your time, and see you all next month in the following webinar. Thank you.
Thank
Bye.
you.